Hi, in today's lecture we are going to discuss another important aspect in molecular biology, that is nothing but genetic code. So before going to genetic code, all of you know what is central dogma, right? What is a central dogma? In which a DNA will undergo a replication process, or DNA will give rise to DNA. That particular process is called what? A replication. DNA will give rise to RNA. That process is called what? Transcription. RNA will give rise to what? A DNA. That process is called what? Reverse transcription. Then this mRNA, DNA will give rise to mRNA, right? So this mRNA will couple with tRNA and rRNA, will give rise to a particular protein. So when I am talking about a protein, what is coming to your mind? The protein is made up of what? Different amino acids. So different amino acids has to be synthesized, and each amino acids has to be bounded together by a peptide bond that will give rise to a particular protein. Okay, so who direct the synthesis of a particular amino acid? That is nothing but our genetic code. So what is the genetic code? The genetic code is the sequence of nucleotides in DNA and RNA that determines the amino acid sequence of the protein. Okay, so this is the nucleotides which are present. So what is a nucleotide? So recall what is the structure of the DNA. So what is the structure of the DNA? The DNA is composed of three major components: deoxyribose sugar, or the ribose sugar, we can tell it as sugar, right, which couples with various nitrogenous bases which give rise to what? A nucleoside. That nucleoside combines with the phosphate group, give rise to nucleotide. So many nucleotides together, it is called what? Polynucleotide. So that polynucleotide is nothing but your DNA. Okay, so this is applicable in both the cases of what? DNA as well as RNA, with only difference, that is in case of DNA it will be thymine, in case of RNA it is nothing but uracil. So genetic code is nothing but the nucleotide sequence, where this nucleotide sequences are present, which is present in the DNA as well as RNA. What that determines the production of what? A particular amino acid. That amino acid will be incorporated into a protein. Okay, right. So we know that the DNA is in linear structure, right? Even though it is in linear structure, the proteins are not directly produced from the linear DNA stretch. Instead, the DNA will be converted into an RNA, messenger RNA. Right, just now we discussed the process is nothing but what? Transcription. So the DNA will be converted into mRNA. So the mRNA will determine the primary structure of your protein. Right, so in case of mRNA, what will be there? The DNA and RNA is almost same. Only difference is what? There it is deoxyribose sugar, it is ribose sugar. And in case of the pyrimidines, in case of DNA it is thymine, in case of RNA it is nothing but what? Uracil. Okay, so in the mRNA there will be the nucleotide stretch A, U, G, C, G, C, U, like that a nucleotide stretch will be there. So the three adjacent nucleotides constitute a particular unit. That unit is called what? Codon. Okay, for example AUG, CUG. So three different adjacent nucleotides will denote a particular, that particular unit is called what? A codon, which will determine a particular amino acid. So each codon will determine, or each codon will direct a particular amino acid to be incorporated into where? Into the protein stretch. Okay, for example the sequence AUG. Okay, so the sequence AUG codon that specify the amino acid methionine. Okay, likewise each nucleotide sequence, the triplet nucleotide sequence will give rise to a particular amino acid. So there are totally 64 possible codons are there in the genetic code system. Okay, out of which 3 are having importance in case of termination. We might have studied in the previous class, we have discussed about termination of translation. In that you might have seen that there are 3 codons like Ampere codon, Ocpel codon and Ocker codon. So these 3 codons does not code for a particular amino acid, but it plays a very important role in the termination of the translation. So the remaining things are nothing but 61 codons are present over there. So this 61 codons will specify 20 different amino acids that makes up a protein. Okay, 3 are not in the picture because they plays important role in the translation. The remaining 61 codons which specify 20 different amino acids. Okay, for example in the previous slide we have seen AUG. AUG codes for which amino acid? Methionine amino acid, which is a very important codon. This is the initiator class of amino acid which will be incorporated in the initial stage of the protein synthesis. The first one will be, in case of the prokaryotes, it will be formyl methionine, that is methionine will be added with a formyl group. In case of eukaryote, it will be methionine. Okay, So the first amino acid which will be incorporated in the translation process is nothing but what? The AUG codon. It means that it is found in the beginning of every mRNA. So the genetic code is universal, actually speaking. Means there are minor differences which are there, which we will discuss in the later stages. That is totally, if you speak, that is the genetic code is nothing but universal. It means that which is identical in almost all species. Okay, with the same codon specifying the same amino acid, whether it is U or a bacteria, that particular codon will produce a particular, the AUG means it produces what? Methionine for U also, it produces what? Methionine in case of the bacteria also. I just generalized the statement. So the great work of genetic code, the proposal of genetic code was given by three important scientists. You might have heard about them, that is Nirenberg, Hawley and Hargobind Khorana in the year 1960s. These are the three pioneers who described the genetic code in case of both the prokaryotes and eukaryotes. So this is the table for the genetic code. You can just see that it looks very complicated, but it's very simple to write. Even in examination also you can write this one. That is, see, what you have to do is, you might have studied about Punnett square, about tall versus dwarf, TT into TT. The same way, you just put a square in that, in the left hand side you put UCAG. Okay, UCAG in one side, and in the opposite side, means just like your Punnett square only, you just put the same sequences in the other side also, UCAG. Okay, you can see in the blue color, that is UCAG at down as well as in parallel way it has been written. Okay, now you have to produce the genetic code. Okay, so how will you produce the genetic code? See how many columns should be there? 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, like that. There are around 16 columns you can find over here. Okay, there are 16 columns which are present there. Now you have to put this. How will you put that? U into U. What you will get? UU. Okay, put the same UU 4 times in each column. So why 4 times? Because I told you that there are 64 amino acids are there, 64 codons are there, right? So 64 codons, totally how many are there here? 16, right? So 16 into 4 is equal to 64. Therefore, to 4 different nucleotides, I mean the codons, you have to put it over here. That is U multiplied by U, you will get UU, right? And the last codon in all these 4 codons, I mean the last nucleotide in all these codons will be UCAG. You just see there, UU, in the all, I mean the last codon, you just see only in the last codon, it is UCAG. Getting this? Same way you have to write for the second column also. U into C, UC, U into C, UC, U into C, UC, U into C, UC. And what should be the last 4 nucleotides? UCAG. Okay, likewise you can fill up all the columns so that you will get without any memorizing, you can directly go and write in the examination. This is nothing but the genetic code. Only thing which you have to remember, which genetic code or which codon specify for which amino acid. 2 or 3 workouts, it can be easily done by you. Okay, right. Next, going to the, what are the characteristic features of genetic code. The first important thing is it is triplet in nature. It is not singlet, it is not doublet, it is triplet. That is singlet and doublet codes does not adequate to code for 20 amino acids. Therefore, it was pointed out that triplet code is the minimum required. The minimum requirement for the protection of a particular amino acid is 3 different nucleotides. Since it is 3 different nucleotides, it is the genetic code is nothing but triplet. So the first property of the genetic code is nothing but it is triplet in nature. The second one, degeneracy. The code is degenerate. Okay, genetic code is nothing but degenerate. What is the meaning of that? The same amino acid is coded by more than one base triplet. Okay, so that's why it is called degenerate. It is not a particular or it is a fixed one. So the same amino acid can be coded by more than one base triplet. Okay. Degeneracy does not imply lack of specificity in the protein synthesis. Just because it is degenerate, you cannot think that the protein synthesis is haphazard. It is highly specific. Okay, it is one among the property that it is having degeneracy, but it does not mean that it is having lot of confusions during the protein synthesis, because same type of codons are coding for different amino acids. Okay, so nothing like that. It will be highly specific. It is highly monitored by the protein synthetic machinery. So the other feature of the, if you see, coming to the degeneracy once again, the degeneracy can be classified into two: partial degeneracy and complete degeneracy. So what is the meaning of partial degeneracy? The first two, see, when I am talking about genetic code, always you should think about how many nucleotides there, 3 nucleotides. The first two nucleotides are identical, but the third nucleotide of is having a degenerative codon. Okay, first two are same, but the third one is different. For example, CUU. Okay, CU is same, right? And I am taking CUU and CUC. So first two nucleotides are same, CU. But in case of the first one, it is nothing but U. In the second case, it is nothing but what? C, which codes for a single amino acid. That is nothing but what? Leucine. So that's what we discussed, degeneracy. That is the property of what? The genetic code. So we told that there are two classes of degeneracy. One is partial degeneracy and the second one is what? Complete degeneracy. In case of complete degeneracy, what will happen is the third codon will be different in all different scenarios. Okay, so the complete degeneracy occurs when any of the 4 bases can take the third position. Okay, any of the any of the means what are all the things are there? Uracil is there, cytosine is there, adenine is there, guanine is there. Anything can be replaced as the third nucleotide. We will study what is Vogel hypothesis later. So you will be very clear when I am talking about Vogel hypothesis in the later case. Okay, so in complete degeneracy, it is the the third nucleotide can be anything. That is uracil, cytosine, adenine and guanosine, which all codes for one particular amino acid. What is that amino acid? It is nothing but serine. Okay, so it is nothing but serine. Clear with this? So there are two types of degeneracies which will be there. One is partial degeneracy, another one is what? The complete degeneracy. Okay, in case of the partial degeneracy, what happens? The first two codons are same and the third codon can be replaced with 1 or 2 bases, but in case of the complete degeneracy, any of the base can be added into the third position. Okay, the next important feature of the genetic code is it is non-overlapping. The genetic code is non-overlapping means, what do you mean by non-overlapping? It is continuous. There is no overlap between one base over another. A non-overlapping code means that same letter is not used for 2 different codons. Okay, it is not overlapping. Same letter is not used for what? 2 different codons. In other words, no single base can take part in the formation of more than 1 codon. Clear with this? It is a non-overlapping one. What do you mean by non-overlapping? That is the same letter is not used for what? 2 different codons. On other words, you can tell that no single base can take part in the formation of what? More than 1 codon. This is another property of what? Genetic code, which is commaless, which is flow. It is in full flow. There is no comma, there is no full stop, nothing. So the genetic code is commaless and there is no signal to indicate the end of the codon and the beginning of the next. There are no intermediary nucleotides between the codons. Okay, see what is the meaning of that? A, U, G, C, all those things it is going on. No, but there is no indication anywhere that this is the first nucleotide, I have to stop there. This is the last nucleotide, I have to stop there. It's not like that. It is continuous. That is, there is no comma or there is no indicator nucleotides which is present in between the start and end base in case of the genetic code. There is non-ambiguity. This is another characteristic feature of genetic code. Non-ambiguous code means that there is no ambiguity about a particular codon. It means a particular codon will always code for the same amino acid. For example, AUG, which codes for what? Methionine today, and it will not code for some other leucine or serine tomorrow. That is, a particular codon will code for a particular amino acid. There is no room for an ambiguity there. That's why it is, the feature is nothing but non-ambiguity. While the same amino acid can be coded for more than one codon, which we have discussed already. The same codon shall not code for 2 or more different amino acid. Okay, the same thing which is given in a different way. The essence of this particular slide is it is non-ambiguous. Genetic code is non-ambiguous. It means that there is no ambiguity about a particular codon. A particular codon will always code for the same amino acid. So in the beginning of the slide, we have seen that it is universal. I mean, the genetic code is universal for in almost every organism in the entire world. So universality of this code means that same sequence of 3 bases encode the same amino acid in all life forms, from simple microorganism to complex multicellular organisms like our human being, right? It's very clear that is the genetic code is same in all cases, all living system. Although the code is based upon the work conducted on the bacteria. See, all the 3 great scientists like our Corona, all those things when we discussed, they worked on bacteria, Escherichia coli, even though that is which is valid for all other organisms. Okay, so the genetic code applies to all modern organisms with only minor exceptions. I told you that almost it is not complete. There are some exceptions in case of what? Yeast, mitochondria and the mycoplasma. There is some variations over there. We will discuss what are all the variations in the coming class. Okay, the next important feature of the genetic code is nothing but polarity. Right, DNA 5' to 3' direction. It's a particular polar in particular direction. It is. The genetic code has polarity. That is, the code is always read in a fixed direction. What is that fixed direction? 5' to 3' direction. Okay, you can just, I mean, you just go back to the previous slide where in which I told you that there is a table which denotes what? Genetic code. This is 5' enter. Okay, with the other one is what? 3' end. It should be always, means if you are writing UUU, it should be read as UUU. Right? I will take another example so that it will be clear. UUG, it should be read as what? UUG, not GUU. Because the genetic code, another important feature of the genetic code is nothing but for polarity, and it should be read in a particular direction, that is 5' to 3' direction. So it is apparent that if the code is read in the opposite direction, it would specify 2 different proteins, since the codon would have reversed the base sequence. Okay, don't confuse with this particular statement. So always the genetic code has to be read in a particular direction because it is having a particular polarity, that is 5' to 3' direction. Okay, so these are all, I mean, these are all the major salient features of genetic code. It is a very important 15-mark question. You can refer the previous question paper. So in most of the years, either translation or genetic code is asked. If the genetic code is not asked in 5 mark, they might have asked for 15 marks, means vice versa, 5 marks or 15 marks is a very important question. So in this particular thing, you have to write what is genetic code before writing genetic code. Before writing any 15-mark question, first you just give a introduction about that particular topic. We don't go directly into that particular topic. I just told about centered dogma, about protein, all those things, then come to the genetic code. So what is the salient features? I mean, what is the definition of the genetic code, the discovery of the genetic code. If you are able to put up, if you try to put up this particular table also, I told you how to put this. There is no need of memorizing it. It's just like your Punnett square. Then write the different features and one paragraph regarding each features of genetic code. So we know that what are all the features? It is triplet, it is having degeneracy, and there are 2 classes of degeneracy. What are the 2 classes of degeneracy? One is nothing but partial degeneracy, in which only the final base will be changed, and complete degeneracy, any of the 4 bases can be included there. That is, it is non-overlapping, and it is commaless, and it is non-ambiguous, and it is universal. It is universal in case of both prokaryotes and eukaryotes, and it is having a particular polarity. Okay, so that's all about the genetic code and its salient features. Thank you.